Hello, my name is John Moyer, and I want to welcome you to the NIH Office of Disease Prevention's Methods Mind the Gap webinar series. This series explores research design, measurement, intervention, data analysis, and other methods of interest in prevention science. Our goal is to engage the prevention research community in thought-provoking discussions to promote the use of the best available methods and to support the development of better methods. Before we begin, I have some housekeeping items. You can submit questions during the webinar via the Q&A pod. You'll need to open the pod by clicking on the Q&A icon. We will open the floor to questions at the end of the presentation. The video recording will be posted on our NIH ODP YouTube channel in a few weeks. At this time, I'd like to introduce Dr. David M. Murray, Associate Director for Prevention and Director of the Office of Disease Prevention.
Thank you, Dr. Moyer. Today's speaker is Dr. Bin-Kai Wang. Dr. Wang is an assistant professor of biostatistics at the University of Michigan School of Public Health. He specializes in randomized control trials, cluster randomized trials, and causal inference, with a focus on covariate adjustment, repeated measures analysis, and pragmatic trial design. His research develops rigorous and practical statistical methods to improve the efficiency, robustness, and interpretability of clinical and translational studies. Dr. Wang's work leverages modern analytical tools, including machine learning, large language models, to address key challenges in trial design and analysis, with an emphasis on methods that are both theoretically sound and implementable in real-world settings. It's my pleasure to welcome Dr. Bin-Kai Wang.
Thank you, Dr. Murray, for this introduction and also for inviting me to present my work here. Hello, everybody. Today, I'm happy to present my recent work from working models to reliable evidence, robust and efficient analysis of cluster randomized trials. So in this talk, I will focus on cluster randomized trials. So in cluster randomized trials, we randomize clusters such as hospitals, clinics, or communities, but not individuals. The advantages of this design, it is because it is more pragmatic, cost efficient, and in many situations, it is the only feasible implementation. For example, if the intervention is a policy. And because of these advantages, it has been increasingly popular in the past 10 years. So we can see the figure shows an increasing number of publications in the last 10 years. However, this design also brings many challenges. Most of them are statistical challenges. For example, we need to deal with within cluster correlation. So in such a design, we believe people within the same cluster are correlated, and our statistical inference must address this correlation to avoid erroneous or undercovered, inflated type 1 error. Another challenge is the estimand. So how do we define the thing we want to estimate in this design? And also model misspecifications. What if the model we choose is wrong? Then do we still have valid inference? So let's consider a motivating example called PPACT. So in this design, the intervention is cognitive behavioral therapy for chronic pain. And in this trial, 106 doctors are randomized in one-to-one ratio to receive treatment or control. So here, each doctor represents a cluster associated with their patients. And the primary outcome is a pain scale, which can be regarded as a continuous outcome. So in this trial, there are two models used for analyzing the data. One is the linear mixed models. The other is generalized estimating equations. So these two models are the common choices for analyzing cluster randomized trials. One dominant reason is that they both specifically model the within cluster correlation. And given the model fit, people usually use the treatment coefficients as the treatment effect estimators. And this workflow represents a typical analysis for cluster randomized trials. And we call this type of analysis model-based inference for cluster randomized trials. What do I mean by model-based inference? So in model-based inference, we start from the data. And then we came up with a model to fit the data. And then from the model, we interpret the related model parameters to answer our scientific questions. For example, in cluster randomized trials, the data is gathered for cluster randomized trials, and the model we use can be the generalized estimating equations. And from this model, we extract the treatment coefficient as the estimator for the treatment effects. So this framework is classical and simple and also commonly used. However, it has two potential issues. The first issue is that the model can be wrong, and misspecified models can lead to biased estimates. For example, here we use the GE as the model, but what if GE is wrong? Like the outcome is not normal distributed, the outcome is nonlinear in covariance, or there are some interaction terms or complex patterns not specified there. In this case, is the treatment coefficient still meaningful? If we just use that as the treatment effect estimator, do we have bias or inflated type 1 error? So this is a potential issue given model misspecification under model-based inference. The second issue specific to cluster randomized trial is that even if the model is correct, treatment coefficient may target an ambiguous treatment effect due to within cluster correlation. So this has been founded in the 2022 paper. They showed that if like the GE using exchangeable correlation structure, they will have a parameter in the final estimand. As a result, the treatment coefficient will converge to something not only involve the contrast of outcomes between treatment and control, but also involve the correlation parameter. So this will make the interpretation very challenging because the thing we estimate depends on how we estimate the correlation. So this is not a good property for either analyze or interpret cluster randomized trials. Because of these two potential issues, we want to promote the estimand-driven inference for cluster randomized trials. So the estimand-driven inference has been popular and been there for many years, especially for individual randomized trials. And that we think is a good idea for analyzing cluster randomized trials. So in this framework, we start from defining the estimand first. The estimand is the thing we want to estimate. For example, the average treatment effect defined as the contrast of outcomes between treatment and control. For cluster randomized trials, we need to be more careful because there are different meanings of average here. Are we averaging over a cluster or over all person individually? So given the estimand, this is our goal, then we collect the data, which is CRT data, and then we choose model-assisted inference. So model-assisted inference means two things. First, the model is selected to target the estimand we just defined. And second, we want to achieve valid inference even if the model is wrong. So it points out two key distinctions from the model-based inference. First, we define the estimand first, and the models are chosen to target the estimand. But best fitting the data is only a secondary consideration. And the second, model-assisted instead of model-based inference means that we want to achieve a property that the model are allowed to be wrong. If the model is wrong, then we still get valid inference ultimately. Okay? So let me give a concrete example, like toy numerical example to illustrate the idea here. Let's say we have 20 clusters. Cluster 1 to 10 has one individual per cluster, and the treatment effect for each individual is 5. And for the next 10 clusters, we have 10 individuals per cluster, and the treatment effect is 1 for each individual. So the first question is how to estimate the individual average treatment effect. So this estimand asks the question, what is the effect for a typical person? So in this toy example, the effect, we have 100 plus 10 total individuals in this trial. So the denominator is 10 plus 100, and the numerator is the total treatment effects among all the 110 patients. So the individual average effect is 1.4. So if we want to estimate this estimand, we can show that this estimand is naturally targeted by GE with independence correlation structure. But if you use other correlation structure, it will not target this estimand. Only if you use GE with independence correlation structure, you can target this estimand. So that's what we call model-assisted inference. You must align your model to your estimand. Now suppose we may be interested in a different estimand called cluster average treatment effect. The question we ask is, what is the effect for typical cluster? So in this example, there are 20 clusters. So the denominator is 20. In the numerator, we have 5 treatment effects for 10, 1 treatment effect for the other 10. So the cluster average treatment effect is 3. But if we want to target this estimand, the standard GE cannot do this job automatically. It requires additional weighting to target this estimand. So that's exactly the thing we want to develop. How we gonna adapt GE to target a pre-specified estimand. So this example shows the gap. We want to define estimands first and then use methods to target the predefined estimand. But the gap is we don't have many available methods, especially robust methods to do this job. There's a line of work in the literature talking about how to do inference in cluster randomized trials, which are listed here. But the existing methods can target an estimand only in special cases. For example, if there are no covariate adjustment to improve precision, or they assume independence working correlation structure, or assume that within cluster correlation goes away asymptotically, or most commonly, they assume the model is correct. So we think this may be too restrictive. So our essential goal in this project is to develop robust and efficient statistical methods for pre-specified estimands in cluster randomized trials. Okay, so here is a list of our contributions. First, we adapted GE and the linear mixed models to achieve model robust inference for target estimands. So by model robust, we mean the inference will still be valid asymptotically if the models are arbitrarily wrong. So that we call model robust. And our second contribution is to use the powerful tool of machine learning to analyze cluster randomized trials. And the advantage of this machine learning method is that it can help us achieve the maximum power. And lastly, we show how to achieve external validity for partially observed data. So by external validity, we mean suppose in a cluster randomized trials, like say in a hospital, we have 1,000 patients, but we only enroll 100 patients from that hospital. And our inference want to generalize our findings from the 100 enrolled patients to the 1,000 population. So that's how we call like a external validity from partially observed data. But in this talk, due to the time limit, I will only talk about the first 2 contributions in detail, and the third one will be given in the paper. Okay, so in the rest of the talk, I will have 3 sections. The first one is how we're going to do robust inference by using a technique called weighted g-computation, and we adapt from GE and the linear mixed models. And the second component is how we're going to do efficient inference by using machine learning. And lastly is some demonstrations with simulations and data analysis. Okay, to begin with, let's talk about robust inference with the GE and the linear mixed models. The first question is why we want to work on GE and the linear mixed models? The most important reason is that this is mostly used in practice. According to a survey, is used by 68% of cluster randomized trials. And it's commonly used because it is easy to implement, and it also has good performance with small to moderate data size. So we want to adapt those familiar models to target certain estimands directly. And the gap here is model-based GE and linear mixed models cannot target certain estimands directly. So we want to adapt those models to achieve the estimand-driven inference. Okay. The method we propose is called the weighted g-computation GE estimator. So here's an example of when we use the GE. We can also use the linear mixed model to do this. This estimator has 4 steps. The first step is to fit GE as Euro. Let's say you can use the independence or exchangeable correlation structure. You can adjust for covariates in pre-specified forms like those prognostic variables. And in this model fit, please make sure to include intercept and the treatment indicator. So this is essential for achieving model robustness. And in the second step, after we got the model fit, we will perform the weighted g-computation. And this weighted g-computation will help us estimate group means, the treatment group mean and control group mean. Here I use mu hat to denote the group means. And in step 3, we will combine group means for treatment effects. For example, if we want to target risk ratio for binary outcomes, the estimator will be the ratio of group means. If we want to estimate the average treatment effect between treatment and control, then the estimator will be mu hat treatment minus mu hat control. And finally, step 4, which is also a critical step, is to use sandwich variance estimator for uncertainty quantification, and the final inference will be based on Wald type inference to build confidence intervals. You can also use T distribution for like in small sample size. Okay, so this is the overall idea of this estimator. So then haven't talked too much about this weighted g-computation. So this will be in the next slide. Okay, so what is weighted g-computation? So this is probably the most technical slide in this presentation, so bear with me. There are some technical details. Let's say, suppose there are M clusters in total, and each cluster I has a population of Ni individuals. Like Ni can be very large, but we only enrolled Mi individuals into this trial. Mi is no larger than Ni. And for each individual, we use the index J. And this weighted g-computation can be is shown in this blue formula. So if we want to estimate the cluster average group mean for treatment or for control, we do this computation. So in this computation, we first make model predictions for everybody under treatment or control. So we assume that person is under treatment. We have the model fit, so we can make this prediction for everybody and for both treatment groups. G-computation is just making that prediction and take average of everybody. So this part is the G-computation. We take average among all observed individuals in that cluster. And the weighting is basically a weighted average of G-computations. So here, because we are targeting the cluster average group mean, so we apply equal weight to each cluster. So you can see the weighting is simply an average. Okay, but if we are estimating the individual average treatment effect, the group mean has another weighted g-computation formula. You can see that the g-computation remains the same, but the weighting now is different. It put equal weights on each individual instead of each cluster. Okay, so to summarize, the weighted g-computation has 2 components. G-computation is just making predictions and take average. This is a classical technique in causal inference for estimating marginal treatment effects. And the weighting is used to target the pre-specified estimands. Here, we just used cluster average and the individual average treatment effects as an example, but in practice, you can target a range of estimands using this technique. And this weighting is used to target pre-specified estimates. Okay, so with this technique and the previous defined estimators, we have some good properties for the proposed estimators. Basically, we have several recognizable model robust lengths. Model robustness is a strong property, right? So you allow the models to be arbitrarily wrong, but it is not automatic. We identify a few conditions that if any of them is true, then we have this good property. So for simplicity in this talk, we assume the observed cluster size is a random number. And the weighted g-computation with GE is model robust under any of the conditions among A, B, C, or D. Let's start from A. So if GE use a correct mean model, then the result will be model robust. So of course, this is it's not that strong, right? So we assume a correct mean model, then the robustness is only about the correlation structure. But it gives you some flavor that if you specify the mean model correctly, then you don't need to worry about how you specify the correlation structure. Even if it's wrong, then you still got valid inference. But you may say if this condition A is too strong, we never know whether the model is correct, right? So then we can go for condition B. If we use a Gaussian working model, then the result is robust. By Gaussian working model, we means identity link function and constant variance parameter in the GE model fit. If you use this, magically, the result will be still valid asymptotically if the GE model is arbitrarily wrong. By arbitrarily wrong, you can misspecify any component, the correlation structure, the outcome distribution, anything. And an alternative condition is C. So I suppose you don't want to use a Gaussian working model. I have binary outcomes. Now I want to fit a logistic regression, so it's no longer Gaussian. But if you fit a logistic regression, condition C says if you use an independence correlation structure in GE, then you're still good. You will still got a model robust property. So in this case, the independence correlation structure will simplify the things, and you can use either logistic regression, Poisson regression. That's all good. We still got this good property. And lastly, another condition is that no individual level covariate is involved in the model. So if we satisfy that condition, which means the GE model just use all the cluster level covariates, which is same for everybody within the same cluster, then the result will be model robust to arbitrary misspecification. In that case, we are just kind of equivalently fitting a model like a cluster level analysis without using individual level data, and we have this strong property. Okay, so this result is our key result, one of the key results in the paper. It gives you the conditions under which we can make the GE inference more rigorous and more robust. But it doesn't need all of those conditions. You just need to pick one and apply that one in your analysis. If I have to recommend one, I would recommend using condition C. Like I think the cleanest method is just to use an independence correlation structure in your analysis. You may ask, oh, how do I account for the within cluster correlation then? So within cluster correlation is an issue, but it's not an issue for bias. It's only for variance. But the variance is automatically accounted by using the sandwich variance estimator. So that's the beauty of this method. We don't need to explicitly model the correlation, but we use the sandwich variance to account for correlation. Okay, so that's for GE. Now, how about linear mixed models? Linear mixed models will have a similar model robust results. The main reason is that linear mixed model can be regarded as a special case of generalized estimating equations. The cluster level random effect is the counterpart of exchangeable correlation in GE. So it is a special case. And everything remains the same. The weighted g-computation and the inference remain the same as GE. And since linear mixed model use a Gaussian working model, which is condition B in the previous result, it is a model robust automatically for both estimands. However, important note is that here we only talk about linear mixed models, but we don't talk about generalized linear mixed models. The difference is that generalized linear mixed models will have a link function, and the random effect will be put inside the link function. So if we have this link function, like a probit link function, it is no longer model robust in this way. So we need additional steps to marginalize the random effects to achieve to target the estimand we define. So this result is put in a separate paper. We can talk about this later. Okay, to summarize, so far we talk about how to do robust inference to target the estimand we defined. We adapt GE and the linear mixed models with a weighted g-computation to do this. And we also show when the inference is robust to the model misspecifications asymptotically. And also, in practice, we recommend adjusting for prognostic covariance. It can usually improve precision. And to facilitate implementation, we develop an R package, MR-STD CRT, to implement the proposed methods and also the generalized linear mixed model methods. Please feel free to try this out. And if you have any questions or found any issues, please let us know. We are happy to address that. Okay, now let's move to the next part, which is how we do efficient inference with machine learning. So this part will be relatively short. Why we want to talk about machine learning? Because the result in the previous section is about the robustness, is to improve the rigor, but it's not to improve the power or efficiency. The machine learning can be a useful tool here because it can capture the nonlinear relationship between outcome and covariance or heterogeneous treatment effects. If we model the data better using machine learning, we may probably have a better power for the trial and ultimately may reduce the required sample size for a planned trial. But this interrogation is not automatic, right? You cannot just replace linear regression with machine learning. This will cause some bias because machine learning is unstable. So to address this issue, we use the standard but advanced statistical tool called efficient inference function to incorporate machine learning into the analysis of cluster-randomized trials. So here is a very high-level technical overview. So we start from deriving the efficient inference function for our estimand in this setting. And then from the efficient inference function, we extract the related nuisance functions. And we will estimate the nuisance functions with machine learning plus cross-fitting. So here cross-fitting means cross-validation. We separate the training and the prediction so that machine learning does not overfit or provide in-sample predictions. And then given the model fit, we can construct nuisance function estimators, and we put those estimators back to the efficient inference function to get efficient estimators with asymptotic variance. And our asymptotic guarantee is that if all nuisance functions are consistently learned with rate m to the quarter, then the resulting machine learning-based estimator is consistent and reaches the efficiency lower bound. So here, by efficiency lower bound, we mean it is the highest power among all regular and asymptotic linear estimators. And here we have some conditions, right? Consistent learn with a certain rate, and this rate can be achieved by several good machine learning methods like random forest, deep neural network, boosting, and others like highly adaptive lasso. Okay, so this result gives you very good theoretical protections, but in practice, how are we going to use, when to use machine learning algorithms? Here is our general recommendations. So first, we recommend using machine learning algorithms when we have relatively large sample size for stable performance. So large sample size is relatively vague, but we would suggest like 100 clusters to start with. Like in our motivating example, we have 106, then you can apply this stably. And also when we detect nonlinear outcome covariate relationship, or we anticipate there are some strong treatment effect heterogeneity, then machine learning methods may be better than just a linear regression to capture such complex relationship and thereby improving power. But overall, we want to say that machine learning is a tool and is an advanced tool compared to classical regression, but it doesn't mean it is an automatic upgrade from linear regression. It depends on the scenario. It depends on the data generating distribution. And we hope this provides you an extra option when analyzing data. Okay, lastly, some demonstrations. So we start from the simulation. So in this presentation, I only present 4 scenarios. The first 2 scenarios are for continuous outcomes. The scenario 3 and 4 are for binary outcomes. In scenario 1 and 3, we will have a smaller sample size, 30 clusters. But for scenarios 2 and 4, we have 100 clusters. And here, the data generating distribution is nonlinear outcome covariate relationship, and also the data within clusters are correlated. And here is the result. Here, ML means machine learning based estimator, and the GEG means the weighted g-computation based estimators based on GE. These are the 2 estimators we just introduced. The left figure shows validity. It gives you the number, the coverage probability of 95% confidence interval. So ideally, we should see 95% in every cell, but here we see all the numbers are close to 0.95, which means it has good coverage probability. And the right figure shows relative efficiency compared to unadjusted analysis. So by unadjusted analysis means the same method, but we involve no covariate. And if the number is bigger than 1, it means using the proposed method with covariate adjustment can improve precision. You can see that all the numbers in the right figure are bigger than 1, which means covariates can indeed improve precision. And we also see that the machine learning have, by using machine learning, we have some really big numbers, like 14. This means it really improves precision a lot compared to parametric regression GE. Of course, this is by design. We use a scenario where we have a non-linear relationship so that machine learning can outperform regression models. Okay. Next is some data applications. We apply the methods to 3 cluster randomized trials. The first one is PPACT, the motivating example, where we have 106 clusters with 1 to 10 individuals per cluster. The second one is work, family, and health study. This study is done in Michigan. It evaluates workplace intervention to reduce work-family conflict, and it has 56 clusters with 3 to 50 individuals per cluster. And lastly is improving rational use of artemisinin. The intervention is malaria test with vouchers of artemisinin provided for positives. This trial has 32 clusters with 39 to 129 individuals per cluster. You can see that these 3 trials span 3 different areas and with different sample size, and we want to demonstrate our methods on the 3 datasets. Okay, so here are the results. There are a lot of numbers. So we apply the proposed methods, the ML and the GEG for each study, and for estimating both class level treatment effect and individual level treatment effect. And the numbers here represent the point estimate and the 95% confidence interval. Because this is just a data application, we are not able to evaluate the validity, whether it has bias or not. But what we can compare is efficiency, whether one estimator is better than the other in terms of power. here we want to highlight the relative efficiency column. Here, the relative efficiency means like a power compared to unadjusted analysis. All the numbers are bigger than 1, which means using covariate adjustment can really improve precision in practice. But we can see that machine learning seems not so like a advantage, brings too much benefit compared to parametric regression. This is also constantly observed in data, in real data. Sometimes it is better, like say in this example, it is 2.22 is better than the GE, but in other situations it may not. So it really depends on the data use case and situations. Okay, so based on the data applications, we want to provide the following recommendations for practical use, for practical analysis of cluster randomized trials. The first recommendation is to name the estimand first. So define it before we introduce any model. For example, it can be cluster average treatment effect. And then we choose a working model that targets it. For example, the weighted g-computation estimator with the correct weight for targeting each estimand. And then we also highly recommend adjusting for prognostic covariates. This has been shown in our demonstrations that it can really improve power and help you save sample size. And finally, it's very important to account for within cluster correlation in inference. So you don't need to model it explicitly, but you can just use variance, the robust variance estimator to account for within cluster correlation. Okay, to summarize, in this work, we proposed robust estimators for estimand-driven inference based on GE and the linear mixed models. And additionally, we proposed machine learning-based estimators to maximize efficiency. Of course, there are many more results and methods in the paper, including how to address cluster-dependent sampling, what are the formal technical statements, and what are the conditions to establish external validity. Okay. Before ending this talk, I want to briefly talk about a recent platform I developed. It may be useful resource for you. The platform is called RCT Bench. It is a public collection of 125 randomized clinical trial datasets. I developed this database for open and reproducible research. So the good thing about this database is that it has 48,000 individual level data on various outcome, treatment, and covariates, and it spans a diverse therapeutic areas. The key here is that all data is clean and free. You can download them and use it for your research, for benchmarking methods, for teaching, for trial design. You can do, and all the trial are free. They are all shared by the original research team for research use, and you can also use it for your purpose. And I also want to spoil a little bit because currently we are cleaning in the final stage of cleaning cluster randomized trials. And we got 54 from all public resources. And we will post those 54 individual level trial data for these trials soon on this website. And you can access this website from this link or scan the QR code here. And here is a screenshot of this website. You can just access this page. You can browse all the trial level information, like the trial number, the paper, the area, sample size, outcome, and you can directly download the files. And also each trial is associated with a clean defined data dictionary. All right. All right, here is a list of references and experts related to cluster randomized trials. All the authors here are experts for cluster randomized trials. And that's all for me. You can access, this paper is co-authored with Chan Park, Dylan Small, and Fan Li. So they are valuable collaborators. I cannot get those results without their help and support. And here is the paper link. And if you have any questions, please feel free to email me at binkai@umh.edu. and I'm happy to take any further questions. And thank you.
Thank you very much, Dr. Wang. We have had questions coming in almost from the outset. And so let me move into those and let you get started dealing with them. One question that came up very early referred to slide 5. Can you go back to slide 5 in your slide set? It might help.
Okay.
I realize it's practically the beginning. There we go, 5.
Mm-hmm.
So the questioner asked why the individual average treatment effect can only be targeted by GEE with an independence correlation structure. The questioner thought GEE can use different correlation structures, but that would only impact the standard error, not the point estimate. Could you clarify?
Yeah, that's a very good question. And this is also a finding by not by me, but from existing work here, this paper, Wang et al. Because like what the question is super intuitive, right? Because correlation should not cause any bias, but it only contribute to the standard error. But unfortunately, for cluster randomized trial, that's what we observe. If you do the calculation, you will see that the estimate of treatment effect coefficient will converge to something. Then that thing is not only the contrast of outcomes, but it also involves some parameters in the correlation structure. If you use the correlation structure, like say, with exchangeable correlation structure, that parameter will factor into that limit. And as a result, we can only use independence correlation structure to target this estimand. But if you use other correlation structure, it will make things very complicated. So that's my short answer. But if you want a detailed explanation, check out that paper, okay, in Contemporary Clinical Trials, 2022.
Thank you. One of the viewers asked, what does model efficient mean? Does a more efficient model mean that the model can have more power for the same sample size?
Yeah. So sorry, I didn't clarify that enough. So efficiency, power, precision, they usually refer to the same thing in randomized trials. So by more efficient, we mean more powerful. We mean more precise. Like the precision is about the estimation, right? So the variance is reduced. We mean more precise. And because the variance is reduced, we mean the method is more efficient in, like say, when we use the same sample size, a more efficient method can yield more powerful test. And also for the same power, it can reduce the required sample size. So they are relatively the same meanings. Yeah.
Thank you. You've suggested in your presentation that we should use estimands to guide the analysis for a parallel cluster randomized trial. How do we choose the estimand for the analysis? Is there a list somewhere, possible estimands that shows the appropriate weighting scheme for GEE or linear mixed models?
That's a very good question. So let me go to the references. I know I have referenced some. So here, if you check out paper number 10 by Kahan, I think he was also a presenter of this series before. He wrote a guidance about how to choose estimands in cluster randomized trials, choosing analysis that answer the right question. My short answer is the estimand should be driven by the scientific question, whether you want to ask ask questions about individuals or about clusters. That determines the estimand. Once we have the estimand, our method will help you choose the methods, like which weighting you should use for to target the corresponding estimand.
And related to that, do you have guidance as to when to choose individual averaged effects versus cluster averaged effects?
I think my short answer is still is case by case. Like say, suppose I want to study a vaccine, right? So it may be more reasonable to to target the individual treatment effect because we ultimately want to know how it works at individual level. But if we want to evaluate a policy like a new like a masking policy, right? So then probably intervention, the cluster average effect, like say on different regions or different schools, different hospitals may be of interest. I think the dominating factor should be the scientific question. So I cannot say generally you should do this in this situation, but I think it's case by case.
If you can go to slide 21. Okay. One of the members of the audience asked, what are we trying to estimate here?
So that's a good question. I didn't talk about it. Here is about cluster average treatment effect. It's only for demonstration. This part of the simulation results is for cluster average treatment effect. For individual average treatment effect, the results are kind of similar. Yeah, thank you for asking that. Yeah.
If we use an independence working correlation, and you recommended that because you said if we do that, we'll have a robust estimate. So if we use an independence working correlation, can we include individual level covariates and still have a robust model?
Yes, exactly. This is a good question. If we do that, I think this is related to this result. If we use independence correlation structure, that's the only requirement. We can include any covariates in the model, like say baseline outcome, age, gender. You can do everything about the mean model. You can fit the logistic regression. You can fit the Poisson regression. That's still good. The only requirement is just a independence correlation structure.
Thank you.
Mm.
You've used machine learning in place of a standard regression model to try to improve precision. When is that likely to actually work? And how many clusters would you need, say, in each arm? Slide 24 suggested that machine learning was not consistently more efficient, whether the number of clusters was large or small. So can you offer any guidance as to when machine learning is likely to help?
That's a very good and tough questions. So let me put it this way. I think machine learning gives you an option to deal with complex data generating distribution, but it doesn't guarantee efficiency gain. And actually, in another empirical data analysis, we found that machine learning in randomized trials in general, may not be as good as general, like classical regression models. So if I have to provide a one sentence response, is that choose regression instead of machine learning. But here we propose the machine learning methods because it gives you an option. In some situations, also in some of our data evaluations, it indeed provide help. It helps when we have a large sample size, we have nonlinear relationship, and there's no gold standard like saying that if you have more than 100 sample size, you will definitely get this. There's just some observations. Like the larger sample size, the machine learning will be more stable, and if the data generating distribution is more non-linear, then machine learning may fit the data better. But I would say it's just as I said here, it is just an option. It's a powerful tool, but it's not an automatic upgrade. For practical use, I would start from classical regression models.
Yeah. Now, in most clinical trials, Dr. Wang, the investigators have to specify their analysis in advance.
Yes.
In advance of looking at the data. So that last slide that you showed us where it wasn't clear that machine learning was systematically better, regardless of the sample size. Here, for example, machine learning is worse in p-packed, even with a large number of clusters for the cluster level effect. It's a little bit better for the individual level effect. And then down at the bottom for X, it's better in both cases. So it's not consistent. If we have to specify the analysis in advance, it seems like the prudent thing to do would be to use the standard regression model rather than taking a chance on something that might not do as well.
Yep. I totally agree. So
Okay.
yeah, I totally agree with you. Go with standard regression in practice and use machine learning unless we detect strong signals that it's gonna work. Like by analyzing historical data, we say, oh, this is definitely better, so let's do this. This needs strong evidence, at least at the current stage. And I'm not sure, like in 20 years later, like people may have better trials, better data, so machine learning may be better. But at the current stage, machine learning is just an option.
Okay. Do you think that the weighted g-computation method will perform well even with a small number of clusters? For example, 10 in each arm or 8 in each arm.
Yes, yes, that's a good question. I didn't show the simulation results here, but in our simulation, it does work well when we even have like 12 clusters. We still have good performance because it is still based on regression models. Like if the GE or linear mixed model works well in 10 clusters, then by applying the additional step doesn't affect its validity. So it's kind of the contingency is on the original model we use. GE and the linear mixed model. If those works well, then we're good. But
Okay.
I don't know whether maybe we may have very limited sample size. We may not adjust for many covariates and go for simple methods.
Right. That brings me to a question about covariates. With these methods, does it matter if covariates are at the cluster level or member level? With standard linear mixed models, there's a cost in degrees of freedom for cluster level covariates, but not for individual level covariates. Does that same scheme apply with the weighted g-computation approach?
Yes, yes, that's a very good question. The same scheme still applies here. And based on my experience, so the theory doesn't matter, because we are doing asymptotic theory, right? So in large samples, you can include both individual and cluster level covariates, and they all work well. But in small samples, we do recommend like only pick a few prognostic covariates, like baseline outcome, and like those with strong clinical signals. Do not include too much. This will cause a lot of degrees of freedom, and only include those, like if like a cluster level covariate is prognostic, it may be worth to invest the degree of freedom for adjusting for it. Yeah.
Understood. Thank you. We have several questions about the applicability of these methods to other situations. So for example, can the weighted g-computation approach be used for binary or count outcomes?
Yeah, definitely, definitely. So as we show in the result here, you can do any outcome. Like condition B is for Gaussian working model, but if you choose condition C, you can use any outcome, like a count of binary, fit a logistic Poisson regression.
Okay. So the applicability to binary count depends a little bit on the correlation structure, or I suppose you could do it if you didn't have individual level covariates, but the independence correlation structure seems to solve more problems than the other options do.
Yes, yes, that's why we think it is the cleanest method to do
Yeah.
it. Yeah.
There are also questions about other designs. So you've been talking about the parallel group randomized trial. In recent work that we've done here at NIH, we've seen that there's a different cluster design that's much more common than a parallel group randomized trial. We're all familiar with RCTs, randomized control trials with individual level of randomization. Many in the audience may be familiar with an individually randomized group treatment trial where we start with individual randomization, but then the patients or participants become correlated over time because they're getting parts of their intervention in a small group or they're dealing with a shared intervention agent. Have you looked at applicability of your methods to the individually randomized group treatment trial?
That's a very, very good question. And I do investigate a little bit. So the method, of course, not the exact same method, but we can develop similar properties for like model robustness, estimand aligned inference for that design. That's for sure with no problem. And one limitation I found is that I have some trial, but the group is relatively small. Let's say the treatment, like people under treatment will go for different, doctors, right? So the people seeing the same doctor, although they are randomized to different intervention, or they are seeing the same doctors. But the number of doctors is usually relatively small compared to the sample size. In one example, we have only 3 doctors, but more than like 200 patients. In that case, if we want to do like a symptotic theory, it will be a little bit there's some technical challenge, right? Because I don't think it's a good assumption to assume the number of doctors goes to infinity. And but we can do that just for method development purpose. But in practice, that may be not very reasonable. So that's one challenge I can see.
Can these methods also be adapted for a step wedge design?
Yes, that's a very good question. And if you look at the reference 11, so this is for recent paper extending the proposed methods for step wedge design. So step wedge design in general is a longitudinal cluster randomized trials, and every cluster is crossed over from treatment to control. And what we randomized here in step wedge is to randomize the timing of crossover, not treatment or control. So this method can be directly extended to that design, but with one caveat is that in in stepwise design, the estimand is even more complex. It's not only cluster average or individual average. Its estimand is complicated by the exposure time and the calendar time. So to define those estimands, the model should be specifically using the corresponding treatment structure so that we can estimate different estimands, and we need that to be correctly specified. So the short answer is the model is no longer 100% model robust, but it will require certain specifications to be correct. And that part is the model treatment effect structure.
This may have to be the last question. If a trial is proposing to use this weighted g-computation using GEE, what impact would that have on the power analysis? For example, would you have to do a simulation to account for the weighting, or could you apply a more traditional calculation? How would you approach the power calculation if you were going to use this weighted g-computation for the analysis?
That's a very good question. So based on my limited experience, power calculation, like in trial design phase, is a little bit, it's not settled yet, like whether to use covariates. I know like 10 years ago, people don't really use covariates in trial, in power analysis or sample size calculation because we need certain data, right? We need covariate data to really justify that correlation, the correlation level, so that we can see the reduced sample size is real. Otherwise, we are not very like conservative. We always want conservativeness to ensure the finding is solid. But in recent years, we see more and more trials using covariates in the trial design. And in the trial design, when doing the sample size or power calculation, we can like pre-specify the method. Like say pre-specify the like which weighted G computation method we use, and which covariate we use, and which correlation structure we use, and weighting we use. Pre-specify anything, and also pre-specify the software, which software we use, and make sure every result is reproducible. And if we can have some historical data, that will make the justification stronger. And that's all I can think of to make everything reproducible and evidence-based, and also specify the rationale of using that.
Thank you very much.
Mhm.
I will turn the microphone back over to Dr. Moyer to wrap up.
Thank you, Dr. Murray, and thank you to everyone who participated in today's webinar. We will be posting a recording of today's webinar on the NIH ODP YouTube channel in a few weeks. Thank you and have a great rest of your day.
Thank you all for attending.